The overall goal of this procedure is to generate pure Plasmodium falciparum Merozoites in high yield for quantification of their opsonization by human antibodies. This is accomplished by first inhibiting schizont rupture with E64 to generate membrane-enclosed Merozoite structures. In the second step, the free Merozoites are harvested and then stained with ethidium bromide for quantification. In the final step, the Merozoites are incubated with various human plasma samples and THP1 human monocytic cells. Ultimately, the opsonization of the Merozoites by the different human plasma samples can be differentiated by flow cytometry.
The advantages of this technique over existing assays of functional immunity to malaria Merozoites is that intact Merozoites are used and phagocytosis responses are robust and quantifiable.
This method can help answer key questions in the malaria immunology field, such as what is the importance of opsonizing antibodies and phagocytosis for immunity against malaria in vaccine or naturally exposed individuals.
Visualization of this technique is critical, as the timing of E64 is essential to ensure the formation of Merozoites and because Merozoite preparation and enumeration is technically demanding.
After magnetic separation of late-stage P. falciparum trophozoites, assess parasite maturation by fixing a thin smear of parasites in 100% methanol for 10 seconds and staining them in Giemsa for 3 minutes. Examine the stained slide under the microscope to ensure that the parasites almost fill the red blood cells and appear to be in the early